everyone back from the break. So our next speaker is going to be Dr. Vineet Bafna, who I'm happy to introduce. I've known him since my dissertation days when he was on my committee. Dr. Bafna is the core director of our Research and Development Core. He's a professor in the UCSD Computer Science and Engineering Department, where his primary interest is in bioinformatics. He supervises and takes part in the development of algorithms for efficient analysis of large biomedical data sets integrated from multiple sources, with particular emphasis on algorithms to manage and analyze data from massively parallel sequencing technologies.
Thanks, Michelle. Thanks again for inviting me to speak today. And I thought I'd talk about something offbeat, but I hope it'll still be of interest to this audience. But I'll start by just sort of placing my talk in some context. So for the iDASH project, we are interested in building tools, enabling tools for personalized medicine. And I've spoken many times in the symposium in the past. And in the past, we've talked about tools for compression and also tools for querying. And I think I'll sort of begin my talk there, but maybe digress onto a somewhat more specific topic about complex structural variation. So just, we of course know that small nucleotide variations are important. Many of them might be either causal or help mediate diseases, but we also have large structural variations in the genome, and by that I mean we take relatively large portions of the genome, from tens of thousands of base pairs to maybe close to a few million base pairs. And these might, in a specific sample, they might get deleted or inverted and so on. And with recent technologies, we can now begin to detect those. So out here, we have this donor genome. We, of course, don't have the genome, but we have sequenced it, and our sequence information is in the form of fragments. And then we take these sort of paired end sequencing data sets, fragments, so maybe a 500 base pair long fragment with the two ends sequenced, and map it back to the reference. And if they don't map 500 base pairs apart, but discordantly, maybe tens of thousands of base pairs apart, then you can conclude that there's a deletion. And also, in this specific case, you can just count the number of reads mapping to the location, and because this region is deleted in the donor, you'll see a loss in copy number. So certainly we can use clues like this to identify structural variations and then sort of go downstream to connect them with phenotypes that we might observe in the sample. And in the past, we talked about a tool that we had built, genome query language, which is not really for detecting these variations, but if you have if some tool has detected this variation, this is a language that allows us to quickly query and retrieve the evidence that was used to predict this. And it turns out to be a useful tool to just quickly validate the kinds of variations that you might see. And so here's an example of a query that retrieved these discordant reads indicative of a deletion. But it turns out that this sort of simple-minded variations that we are talking about here are not really sufficient to explain all of the complexities of the genome, and this is particularly true in cancer. So here's some example of, this is cell line data, but primary tumor looks somewhat similar, and this is old data. This is array hybridization, where we are really measuring copy numbers, and on the x-axis you see the entire chromosome. And in both of these examples, you can see that there's a tremendous variation in copy number. And so here you can even see, this is on a log scale, there's sort of an exponential growth in the number of copy counts followed by a complete depletion. And these kinds of patterns are very unusual and somewhat hard to explain using only a sequence of these simple or progressive variation. And so in the last few years, people have been trying to figure out what the mechanisms might be for explaining these kinds of variation. And the starting point of our research was this nice paper in 2007, which said that the kinds of variations, this is a very complex data set, but the kind of variations that we are seeing remarkably recapitulates the breakage fusion bridge first presented by Barbara McClintock back in 1941. So a very old story. And the question is, when people look at very complex data and immediately come out with a very exotic mechanism, you almost have to take this with a grain of salt. You have to say, do they really know what they're talking about? And that was sort of the starting point of our research. And here's another example. This one's a more recent one. And it's also quite remarkable. So here, and this is the preeminent group in cancer genomics, Stevens et al., and they presented this paper in Cell a couple of years ago, where they showed a very peculiar pattern of rearrangement. So again, these lines here represent discordant reads, like we had said, and so you can see just with the density that this genome is completely rearranged, just huge amounts of rearrangements. And if you look at the copy numbers, yes, there's a big change, but what's peculiar is that they're oscillating. So the numbers change between 2 and 4, 2 and 4, 2 and 4. So very unusual mechanism of rearrangement, and they come up with a completely sort of, not, I wouldn't say unusual, just out-of-the-world hypothesis. They postulate that the genome just shatters into pieces within a generation and then miraculously comes back together and makes copies of each other. And so when you have hypotheses like these, which, by the way, this paper was highly cited. It has over 300 citations in the last 2 years. And since this paper was published, many people have claimed to find chromotripsis in their work. So one of the things we would like to do is to re-examine these data sets and say, is this indeed a mechanism that is plausible? Because it's an extraordinary hypothesis, and it requires a higher level of proof. All right? So these are the kinds of problems that we have sort of worked on in the last few years. And I won't talk a lot about chromotripsis today, but I will give you some examples of how we are studying this. And certainly I'd like to talk about breakage fusion bridge. All right, so yes, why we should care is very simple, that people subtype cancers in different ways, right? You look at maybe what tissue it occurs in or what kinds of mutations you might see. This is another way of thinking about cancers, is what is the mechanistic reasons for the genome rearranging in the way it is. And understanding and detecting these mechanisms might help us predict the pathogenicity and the progression of cancer and so on. So it's a basic science question, but one of great importance. So let's start with breakage fusion bridge. What is it? And as I mentioned before, it comes from this remarkable paper by Barbara McClintock, who's of course one of the celebrated scientists of our century, but she had the hardest time being a woman in those days to find a proper job, and she moved from place to place. And this was work done when she was at Missouri for a period of 5 years in the late 30s. And so she observed in maize chromosomes that sometimes there's a break in the telomeric region of the chromosome. And when this break happens, the chromosome becomes unstable. So during replication, a bridge is formed to stabilize the ends of the chromosome. And now, as the sort of centromeres move apart, they'll cause a stress on the chromosome, and the chromosome will break again, not probably at the position that it was broken before, but at a different position. So what happens, if you think about the segments of the chromosome arbitrarily labeled as A, B, C, D, E, F, G, then this breakage really left a portion of AB out here. And one way to think about this is that this prefix of the chromosome duplicated and inverted. And now the chromosome again has a broken end, so the cycle can revisit. And sorry, and so this time the break might happen elsewhere, and you might get a rearrangement like this. And in a few cycles, you end up with a completely different and complex rearrangement, right? So this is breakage fusion bridge in action, and she observed this in maize. So why are we interested for tumors? Well, telomere loss is indeed seen in many tumors, and it's sort of a leading cause of chromosome instability. But there's a lot of data now which suggests that this has some role to play in cancers. Here's some examples of the breakage fusion. So we see these dicentric chromosomes, which indicates that there was a breakage fusion bridge formed right in the middle. And in similar sort of painstaking experiments, people have teased together these different aspects of BFB just through cytogenetics. But this is not a mechanism that we can use in a high throughput setting. You cannot really observe BFB in action in tumors that might have been resected from individuals. So if you have to detect this and really understand how often this thing occurs, we have to really go back to the molecular data, which means genome sequencing, hybridization, those kinds of techniques. So what can we do with sequence data? Well, right now we don't have the capacity to take short reads and assemble them, and something that's highly rearranged like this, we have no hope of reassembling in any time. But what we can do is we can take all these fragments, map back to the reference, just like I said in the beginning, and measure counts, right? So copy number data. So in this case, if this was our genome, and we know that this came through BFB operations, we can just sort of count the fragments and say that, well, A appears 4 times, B appears 4 times, and so on. So this is the data that we are given, and the question we are asked is, if you have given copy number data like this, is it consistent with breakage fusion bridge? That is the problem that Bignell et al. In their genome research addressed, and they said, well, we can look at this copy number counts, and we can claim that breakage fusion bridge occurred. The problem is not actually as simple as you might think. A simple example I might give you is you take the same data set, and you change A by 1. And so here there were 4, and here there's 3. Everything else remains the same. While the first one can be explained by breakage fusion bridge, the second one cannot. In fact, there's very interesting combinatorics in the sense that all even numbers must follow all odd numbers. So we can already immediately say that this one doesn't satisfy. But in general, if this is the problem that's given to us, given copy number data to find if it is consistent with BFB, it requires a fair amount of work. When we published our paper earlier this year, it was almost a 40-page proof of how the algorithm works. But I won't go into the details of that algorithm today. I'll give you some ideas of the combinatorics, just and some initial work that we had done, which illustrates some of the nice ideas here. So here, this is not an empty slide. You can see that there's the genome out here. And let's say we apply a breakage fusion bridge operation. So you can see that AB inverted, duplicated and inverted. And what I'm doing is always make sure to write A on top of A and B on top of A, so you can see that it inverted. And that allows us to see some symmetry. So we do this the next time. Now AAB inverted and duplicated, so we get this as the genome. And then one more round out this. And then in the final operation, we take the entire genome and duplicate it, and we get this. And so the data that we really see is not all of this path, but a count of 12 for A, a count of 10 for B, and a count of 2 for C. And we have to ask, from this count, can we construct a path like this, which is explainable with BFB? So that's another way to look at the data. And it sort of gives us an idea of how we might address these problems. So what we can think about is you take the first pair of Cs. If you start from here, and then the first time you see a C again is out here. So join these together. And now do the same for Bs. So now we have 5 Bs, 5 pairs of Bs, and then we have 6 pairs of As. So you can see that there's a nice hierarchical structure. We can think of this thing as being the root node of a tree, and then the tree has 5 children, which is the B level, and then that has 6 children arranged in a particular way, and that's the A level. So we call the object on the right is what we call BFB trees. These are trees which have special symmetries. And it turns out that if those symmetries are satisfied, there's a one-to-one connection between these trees and breakage fusion bridge sequences. All right, so one of the things is that it should have long ends. That is, the ends of the tree should be at least of a depth that's no less than any depth inside. So that's long ends. Also, if you take any pair of any sort of node and rotate the tree around it, you should get the identical tree, so it's symmetric. And if you take any adjacent pair and rotate the induced tree around it, you should get an identical tree. So if it has these 3 symmetries, it forms a BFB sequence, and that gives you gets you part of the way there. So we have an algorithm that's based on this. I wouldn't go too much into this algorithm. It involves the solving of some Diophantine equations. For example, here we have one node. We have to see how to fit 5 children. There's only one way to do it, and we can do that. And now what we see is that there's symmetry out here. This tree must be the same as this tree, so we forget about this and just give these nodes a multiplicity of 2, and then just build the tree on the right side. And so now we solve a different equation, and all the solutions that satisfy these are possible candidates. And after we do that, the multiplicity increases. So because the multiplicity keeps increasing, we don't have to worry about a large portion of the tree, and that speeds up the algorithm. It's still an exponential time algorithm, but it works. To get from here to actually get to sort of linear time, which we achieve, was required substantial effort, and I will not go into that. All right, so hopefully that gives you an idea. And now with this tool in hand, we can analyze different data sets and say if they are or they are not BFB. So this is one example where this is a sequence that's given to us. The closest BFB sequence that would be achieved would be through this count vector. And now, we can see that going from count vector 1 to 2 is difficult because usually our counts are very reliable here. Going from something like 8 to 9 would be much easier because the counts are not as reliable. So in this case, we can confidently say that this is not BFB. However, when we look at some real data, we do find specific cases where where we see BFB. And so I'll just give you some examples. This is on simulated data, and just shows that in a case where we embedded BFB operations among regular rearrangements, we can detect about 80% of the BFB operations at a 5% false positive rate. This is an example from a patient with pancreatic cancer. And here we found a BFB that was already predicted by Campbell and others in Nature. And in the same data set, we identified one more sequence which has BFB, and we reported on this earlier this year. All right. So just sort of to complete this story, we have a collaboration with Tom Reed and NCI where we are predicting BFBs in some of their cell line data sets, and they will do cytogenetic experiments to confirm the validity of these experiments, but those are ongoing. All right, so let's talk a little bit about chromotripsis, and then I'm going to stop in the interests of time. It turns out, at least that's our opinion, that chromotripsis simply doesn't occur because when Stevens et al. Presented their data, they had 5 basic operations, and there were only 2 that were really quantitatively checkable. Everything else is interesting, but not a way to distinguish chromotripsis from non-chromotripsis. So the hypothesis that they bring about is that the genome shatters into many pieces and comes back together. And the opposing to this, opposite of the null hypothesis would be simply that there's a collection of progressive simple rearrangements that gives you the same pattern. So what they say is that even though there's extensive rearrangement in the data, there's only 2 copy number states. And the way they predict that happens is that the chromosome shatters, and the ones with high copy number states come back together. The ones with low copy number states get removed from the chromosome. So now you have one regular chromosome, one hugely depleted chromosome, and then there's a genome duplication afterwards, which would give you these copy numbers. So, but that's not the only way to do this. And we find that there are many ways in which you can sort of say that that's not necessarily the only explanation. First, in their simulations that they did a bunch of computational work to show that what they were seeing was very unusual. There was a little bug in it, and once you fix that, some of the significance goes down. We created a genome, it was a simulated genome. It was created simply through progressive rearrangements, and if you run their tool on it, you can see that it will predict that it's chromotripsis. So now we know that it's something which is progressive, and the tool predicts that it's chromotripsis. And finally, we go the other way also, and we take the one chromosome that they predicted, which had over 200 breakpoints and 2 copy number states, and we came up with a scenario just based on progressive rearrangements, inversions and deletions, which gives you exactly the same data set. So when you have an extraordinary hypothesis like this, they need an extraordinary level of proof, and we believe that this is not yet done. Here's quickly an example from their data. They have these 3 predictions of chromotripsis. In every case, we could predict a bulk of their breakpoints. So out of their 55 breakpoints, we could predict 52, here 74 out of 75, and 228 out of 232. Similar numbers to what they achieved, but using only progressive rearrangements. And the picture on the right shows this is their data. This is what we achieved using only progressive rearrangements. And as you can see, it looks very, very similar. OK, so I'll just sort of leave you with these concluding thoughts. We certainly have a route that we are moving towards, which is correlating the changes in our genotypes with specific phenotypes. And yes, structural variations play a big role. But we must also consider that the genome is a lot more complex than we think in many specific cases. And there are many mechanistic aspects of these rearrangements that are poorly understood, where computation and sort of reanalysis of the data can play a big role. And there are sort of 3 lines of research that people are pursuing. One of them is just that if you're given fragmentary data, can you reconstruct the architecture of the full genome? And we are not quite there yet, but recent advances in technology, for example, the HeLa genome was recently assembled using a mix of different technologies. So it seems like that might be, you might be able to do it for cancer genomes too. The other question is, if you're looking at cancer genomes, can you sort of predict the evolutionary scenario that best explains the evolution of this tumor genome? And with advances in single-cell sequencing and so on, we might be able to achieve that. And then finally, what is the mechanistic basis of these rearrangements? And that was the kind of question that we addressed in the talk today. All right. So thank you very much. And I'd like to acknowledge the few people who worked with me on this project, and some of the publications that I talked about I've mentioned out here. Thank you. Questions? Yes.
So in the progressive rearrangement model, can you estimate how many iterations were involved in that?
Yeah, it's a very, the question that was asked was, can you estimate the number of rearrangements that was caused? Actually, there's a very simple way of doing that, and it doesn't require any kind of machinery. A rearrangement creates breakpoints. So if you have a deletion, it creates a single breakpoint. If you have an inversion, it creates 2 breakpoints, because a breakpoint is where 2 disparate regions of the reference genome come together in the sample genome. If you have a translocation, it creates 3 breakpoints. And you can sort of generalize to anything with k breakpoints. If you have a data set with 200 breakpoints, and you use an operation that uses 2 breakpoints, you need about 100 breakpoints. But if you're reusing, you might need up to 200. So it's between 100 and 200. Now, some people say, well, that's a lot of breakpoints. But we have to imagine that this is a genome which is where certain sort of protective genes have already been deleted, and now it's sort of free to rearrange, and there's selective advantage to doing so. So it sort of gains steam. So it's not hard to think that in a single generation it occurs, it might have 5 or 6 rearrangements, and that means in 20 generations it might have the 200 rearrangements. So we don't think that this is a higher, and we're not saying that it is progressive. We're just saying that the hypothesis that was proposed for chromotripsis of everything happening in 1 generation is even more extreme than what we are proposing. So that's our argument. That was a good question. All right, thank you.